Hello everybody, and welcome to Chapter 3, which is all about the cell. We are going to start this chapter off with a little piece of history of biology, which is not something that I want you guys to know or be able to regurgitate on an exam, but rather is just an interesting story that leads us into the topic at hand, and that story is about this guy named Robert Hooke. You may have not have heard of Robert Hooke before, and we'll talk about why that is the case in just a second. But on the left-hand side of the slide is a picture of what we believe he may have looked like. And I say may because there are actually no surviving portraits of Robert Hooke. And the reason why that's the case is because of another scientist that you almost certainly have heard of, named Isaac Newton. So it turns out that Isaac Newton and Robert Hooke were terrible enemies with each other. They were both competing to be the preeminent scientific mind of their era, which was in the late 1600s. They were both competing to be the president of a scientific society called the Royal Society of London. And in fact, Robert Hooke, for a period of time, was the president, and Isaac Newton had to submit his research to him. And they would squabble and fight in these angry letters about who was the actual discoverer of certain scientific principles. And all this went on until Robert Hooke's death in 1703, when Isaac Newton succeeded him as the president of the Royal Society of London. And he hated him so much still that he had his portrait taken down and destroyed. So that's why we have no surviving portraits of Robert Hooke, because of Isaac Newton. And then Isaac Newton proceeded to basically bash his reputation as best as he could and try to just sort of tamp him into scientific obscurity. But despite this, Robert Hooke was one of the greatest scientific minds of his time. He discovered the scientific principles that made it possible for the first truly accurate clocks and watches to be made. He was the first person to propose that light traveled as both a wave and a particle. But most importantly, for our purposes here, he was able to develop and use microscopes that were some of the most sophisticated microscopes that had ever been created at that time. And he recorded his observations through microscopes in this book called Micrographia, which is the first book ever printed with images of objects seen through a microscope. And so here are some of the images from that book, Micrographia. Up in the left-hand corner, that is a water flea. Down below it is some mold. In the middle is the veins on a plant called stinging nettle. And on the right is a drone fly looking straight on. So these are all very interesting and cool drawings that he created, but probably the most relevant and interesting thing that he found when looking through his microscopes was this. So while looking at pictures, or rather samples of cork tree that he drew pictures of, he noticed that the cork tree was perforated and porous, much like a honeycomb, and this is his own words. And he noted that these pores were the first microscopical pores that anyone had ever seen and anyone had ever written about, because no one had been able to see things under the microscope in this level of detail before. And what he was actually looking at, those microscopical pores that look like a honeycomb in this piece of cork tree, what he was actually looking at were cells. He was the first person to recognize and be able to visualize the fundamental units of life. So in this lecture, we're going to be continuing on with our discussion of one of the principles, one of the qualities that unifies living organisms, and that is their specific type of order. We previously went over the molecular basis for life and the types of biological molecules that compose living things. And now we're going to be moving up to the next level of order, which is how cells are structured in the living organisms. Now, Robert Hooke's observations came long before people had a true understanding of cells, but today we have a much more thorough understanding and much more powerful tools for being able to observe cells. And today we understand that cells are so essential to life that there's this set of 3 tenets called cell theory, which essentially wrap up the role that cells play in living organisms. So here are those 3 tenets. The first one is that cells are the smallest living units. Of course, there are things that are smaller than cells. There are molecules, DNA, protein, carbohydrates, these sorts of things. However, those things are not alive. They are simply chemical compounds. Cells are the smallest units that exhibit all of the characteristics of living organisms. Number 2, all living organisms are composed of cells. So everything that we have recognized in the world that qualifies as being alive is composed of cells. And then last but not least, all cells arise from pre-existing cells. This is something we'll talk more about later, how cells are formed. They are essentially born out of existing cells, and we'll talk about those processes later. Now, believe it or not, everything that you see on this slide here is an example of a cell. You can see clearly that they all look quite different from each other, but they are indeed all types of cells. We have a plant cell up here at the top. Down here, these are bacterial cells, and down here, these are a type of animal cell. And we will talk about the different types of cells that exist and the ways that cells can be different from each other. But before we do that, we're going to start by talking about what all cells have in common with each other. There are 4 qualities or structures that all cells have in common with each other. And the first of these is that all cells have a plasma membrane. A plasma membrane is essentially the barrier that separates the interior of the cell from its environment. It is a thin, flexible sheet that has an important purpose of dictating what can go in and out of the cell. The plasma membrane is composed, as we've mentioned before, of molecules called phospholipids. It's composed of 2 layers of phospholipids that are facing each other, as can be seen in this right-hand image right here. The phospholipid has a unique structure because one end of the molecule, that is the fatty acid tails, are nonpolar and therefore they are hydrophobic. They cannot dissolve in water and want to separate from it. The other end of the molecule, which is the phosphate head, is polar and hydrophilic, meaning that it does like to dissolve in water. So when you get conglomerations of these together, they organize themselves all on their own into this bilayer that composes the membrane because the phosphate heads want to dissolve and interact with water, and the fatty acid tails want to escape from water, and this arrangement satisfies both of them. So all cells have a membrane. That's one thing that they all have in common. Now, as I said, there are 4 different structures that all cells have in common with each other. And plasma membrane is just one of them. But before we get to the other 3, we need to sort of discuss and elaborate on the plasma membrane in more detail because the plasma membrane plays an extremely important role, and that is regulating what goes in and out of the cell. So here's a look at a plasma membrane represented on a more detailed level. You can see the phospholipids represented here, and those phospholipids are shown in these little orangey colored hydrophilic phosphate heads that are attached to the hydrophobic phosphate tails. You see them in 2 layers facing each other. But you can also see that there are these blue structures embedded in the membrane, and those blue structures are proteins. Proteins are extremely important in the membrane. They perform a variety of functions. Some of them serve as channels that are responsible for transporting molecules in and out of the cell. Other ones serve as anchors for carbohydrates that may be identity markers for the cell. And you can see those highlighted in the yellow boxes here. The cell constantly holds these identity marker molecules out on its surface so that other cells can recognize it and determine whether it is friend or foe. And proteins can serve as anchors for these molecules to stick out off the surface of the cell. Finally, proteins that are embedded in the membrane can also serve as enzymes, which facilitate essential chemical reactions. You can see that there are two different types of proteins embedded in the membrane here. Integral proteins, as they are called, are fully embedded in the phospholipid bilayer. Many of them run completely from one side of the membrane to the other, and these proteins are called integral proteins because they are integrated in the membrane. Peripheral proteins, on the other hand, are attached to the surface of the bilayer on either one side or the other. And these ones are called peripheral proteins because if you think about what the word peripheral means, your peripheral vision means at the edges, right? And so the peripheral membrane proteins are just attached to the membrane at the edges. They are not fully integrated or embedded like the integral proteins. And another component of the membrane that we can see highlighted here is something called cholesterol. So cholesterol is located in the hydrophobic part of the bilayer, meaning that it mingles with the fatty acid tails. The cholesterol in the membrane is responsible for keeping it fluid, even as temperature changes. So in other words, as temperature drops and the environment of the cell becomes colder, the membrane might tend to stiffen up and get harder, as we know things become more solid as temperature drops. However, cholesterol prevents this from happening and keeps the membrane fluid. This is only a feature found in animal cells in particular. Other cells lack cholesterol and may have different, separate mechanisms for keeping their membranes fluid. So as I said, the membrane plays an especially important role for the cell because not only does it separate the cell from its environment, it also regulates the passage of materials in and out of the cell. It determines what can enter the cell and what can exit the cell and the mechanism by which those entries and exits can take place. There are three different mechanisms by which things can enter and exit the cell environment, and we're going to talk about each of these three. The first one is passive transport, the next one is active transport, and then finally there is bulk transport. And the name of each one sort of makes sense. As we are going to see, passive transport occurs without the need for any effort by the cell. It happens passively. Active transport will require some energy expenditure by the cell. It has to go out of its way to make active transport happen. And then finally, bulk transport will be the transport of large quantities of molecules all at the same time in a bulk fashion. So we're going to start by talking about passive transport first. Passive transport is more or less synonymous with a phenomenon known as diffusion. Diffusion is the natural tendency of molecules to move from areas where they are highly concentrated to areas where they are less concentrated. And in the definition that you see here, solute, this refers to a molecule that is dissolved, usually in water. We know that living organisms are mostly composed of water. Water is found both inside and outside of their cells, and so therefore molecules in living organisms are essentially synonymous with solutes. Diffusion is a phenomenon that you've probably experienced before. For example, if someone goes in one corner of the room and sprays an air freshener, after a while, those molecules of smell-good stuff are going to start spreading out throughout the room, from that one corner where they started to the rest of the volume of the room. And this is diffusion in its essence. It happens because the molecules want to become less concentrated. They want to spread out and extend the distance between them. This is a natural tendency for molecules to do, and so therefore the cell doesn't have to expend any effort for molecules to move this way. They just do it on their own, as we know. So simple diffusion is one variety of diffusion, where molecules are able to pass directly through the plasma membrane, down their concentration gradient. So in other words, if molecules happen to be more heavily concentrated on one side of the membrane, they can pass directly through the membrane if they are capable of simple diffusion and make it to the other side in order to spread themselves out to the maximum degree. This would sort of be like the equivalent of walking through walls for us. And not many molecules are capable of walking through walls. This is sort of a rare trait. Most things cannot pass directly through the plasma membrane in simple diffusion. But some examples of things that can include the molecule oxygen and steroid hormones. So if most molecules can't walk through walls, how do they get inside or outside of the cell? Well, they use doors or windows. So another type of passive transport, another type of diffusion, is called facilitated diffusion. If simple diffusion is walking through walls, facilitated diffusion is using a door. And that door is made of protein. Like we saw when we talked about the structure of membranes, proteins are embedded in that phospholipid bilayer, and many of those proteins serve as channels or passageways for molecules to move in and out. This is called facilitated diffusion because it is still diffusion. It does not require energy on the part of the cell. The cell is not expending effort to make this happen, but it is being facilitated or helped out by the presence of these proteins since these molecules can't move through the plasma membrane directly. Now, the third and final type of passive transport, which is perhaps the most tricky, is something called osmosis. So, osmosis is a special type of diffusion. We saw that diffusion is where highly concentrated particles spread out to areas where they are less concentrated. Osmosis is sort of the opposite of this. Whereas diffusion pertains to the stuff that is dissolved in water, osmosis pertains to the water itself. And whereas the molecules that are dissolved will move in one direction, osmosis is the tendency of water to move in the exact opposite direction. So osmosis is, as I said, a special type of diffusion pertaining specifically to water molecules, where water will move in the opposite direction of the solutes or the dissolved stuff. Let's take a look at how this might work in an example. So here we have a beaker, and we have two separate sides to this beaker with different concentrations of dissolved stuff. The pink fluid represents the water, and the purple spheres represent the stuff that is dissolved in water. And what this red dotted line right here is meant to represent is a barrier that cannot be permeated by the stuff that is dissolved. These purple spheres on a molecular level are too large to pass through this filter right here. Water can move back and forth, but the stuff that's dissolved in water is too large to move back and forth. We know that the tendency of the purple spheres, based upon diffusion, would be to move in this direction here. They would want to move from the right-hand chamber to the left-hand chamber because they want to spread out. They want to maximize the distance between them. But we also know that they cannot do that. That membrane, that filter, is preventing the solutes from passing through. So instead, to achieve the same end goal, water can move in the opposite direction, from the left-hand chamber to the right-hand chamber, and cause the molecules to become maximally spread out, which is the end goal of diffusion as well. But in this case, it has been achieved by osmosis and not diffusion. Now, this phenomenon, osmosis, has consequences for cells because oftentimes it is the case that while water can move through the membrane easily, other larger molecules cannot. And so osmosis can sometimes have a more dramatic effect on cells than diffusion does. This figure here summarizes the three different consequences that can be experienced by cells based upon osmosis. So in the left-hand panel, we can see something represented called an isotonic solution. The prefix iso means equal, and so an isotonic solution describes a cell that is bathed in a water solution where there is an equivalent concentration of particles outside of the cell as there is inside of the cell. If everything is already equally spread out, both outside and inside of the cell, then there is no particular impetus for water to move in either direction. Therefore, there is not a rush of water into the cell or a rush of water out of the cell. Everything is in equilibrium. The middle panel, this is not the case. The middle panel shows what's called a hypotonic solution. Now, the prefix hypo, if you think about what you already know about the prefix hypo, hypothermia, for example, that means your body temperature is too low. And so a hypotonic solution means that a cell is bathed in a solution where the concentration of particles or solutes there is too low compared to the inside of the cell. If there is a higher concentration of solutes inside of the cell compared to outside of the cell, we know that water wants to move toward the higher concentration, and therefore there will be a swelling of the cell as a result. Sometimes that swelling can be so dramatic that the cell actually completely bursts. Of course, that's not a good thing for a living organism. And on the right-hand panel, we see the opposite scenario. We see a cell in a hypertonic solution. A hypertonic solution, if we think about what we already know about the prefix hyper, hyper means elevated, right? It means too high. And so if a cell is bathed in a solution where it has too high of a concentration of solutes, then the opposite thing happens as in a hypotonic solution. The water wants to move toward that higher concentration, and so it will rush out of the cell, causing the cell to shrink or crenate. So the perfect concentration for a cell is an isotonic solution. Hypotonic is too dilute, hypertonic is too concentrated, and both of those can have consequences for the cell. Now, here's something interesting that you may or may not know. You can drink so much water that you can actually die from what's called water toxicity. The reason why this happens is because of osmosis. When you drink too much water, what happens is you create a too dilute extracellular environment for your cells. You create a hypotonic solution. As a response, your cells start to suck up all of that water that's in their environment in order to create this osmotic balance. And when this phenomenon happens in the cells of a person's brain, this is what can lead to them dying from too much water. They can actually develop symptoms that are similar to meningitis, including extreme headache, nausea, and then later coma and death because their brain cells are swelling because of the hypotonic solution that has been created by drinking too much water. Now, under normal circumstances, this is not a threat to people. You have to drink a pretty significant amount of water for this to be a danger that is presented to you. It has to be more than well over a liter per hour, and most people aren't drinking that much. But there have been some tragic cases of people who are either exercising or they entered a water drinking contest and they've died as a result of drinking too much water. So while it's important to stay hydrated, don't be too hydrated. So that completes our discussion of passive transport. And we're going to move on now to a discussion of active transport, which luckily will be a lot more simple and straightforward than talking about all the complexities of passive transport. We saw that passive transport was the movement of materials in and out of the cell in a way that happened naturally, without any effort on the part of the cell, because diffusion is just a natural phenomenon that is driven by itself. Active transport is not like this. Active transport requires effort on the part of the cell in order to move molecules up their concentration gradient. In other words, in the opposite direction that diffusion would dictate. So for example, we talked about the air freshener, right? We go in the corner of the room, you spray the air freshener, and then gradually, by itself, those molecules spread out throughout the room. Will those molecules spontaneously recollect themselves in one corner of the room? Will they reconcentrate themselves? No. In order to make that happen, you would have to expend energy to collect up all those particles and pack them back into a highly concentrated area. That, in essence, is active transport, moving molecules in the opposite direction that they naturally want to go. Sometimes the cell has to do this. Sometimes the cell needs something and it has to move it in the opposite direction than it wants to go. And when it does this, it expends a particular type of fuel called molecules of ATP, which we'll talk more about later. But that's what you can see going on in this animation right here. We can see these little molecules of ATP being expended in order to pump molecules back and forth across the membrane in the opposite direction that diffusion would dictate. So diffusion does not say that these little yellow spheres should go left and these green spheres should go right. In fact, that's the opposite of the direction they would want to go. And so therefore, the cell has to use ATP fuel in order to make that pumping action happen. And then finally, third type of transport is called bulk transport. We've talked about passive, we've talked about active, now we're talking about bulk. Bulk transport moves materials using these pockets called vesicles. Vesicles are little loops of membrane material that can bud off of the existing cell membrane in order to bring particles in, or they can merge with the cell membrane in order to dump particles out. And we have two separate names for either the moving in or moving out action of bulk transport with vesicles. Endocytosis is the movement of substances into the cell using vesicles. And here again we see that prefix cyto, which means cell. Endo means inside, cyto means cell, so endocytosis is movement inside of the cell. The opposite of this is exocytosis. Exocytosis, exo meaning out, exocytosis means the movement of substances out of the cell, using these pockets of membrane material called vesicles. So finally, we've reached our first checkpoint of the lecture here. We have an animation here showing a number of different substances crossing the membrane, and I want you in this checkpoint to focus on the blue spheres. By what transport mechanism are the blue spheres crossing the membrane? And be as specific as possible as you describe this. Next, in this checkpoint, I want you to tell me by what mechanism the orange spheres are crossing the membrane. And again, be as specific as possible. And then finally, checkpoint 3, tell me by what mechanism the pink spheres are crossing the membrane. Now, we just spent a lot of time dwelling on the plasma membrane in particular, but recall that the reason why we were talking about the plasma membrane in the first place is because it is one of the structures that is common to all types of cells. I said that there were 4 structures that were common to all types of cells, and now we're going to mention the other 3, although we're going to go into far less detail about them than we did about the plasma membrane. The second structure that is common to all cells is something called the cytoplasm. Again, we see that prefix cyto here. So cytoplasm is the semi-fluid substance that fills up the inside of the cell. It's a little bit more viscous than water, a little bit thicker and more syrupy than water, but it's basically the stuff that fills up any sort of dead space that is inside of the cell. Next is genetic material. So all cells have DNA that encodes their traits and that allows inheritance to happen by passing down those traits to offspring. And the fourth and final characteristic that all cells have in common is they all possess ribosomes, which are clusters of RNA and proteins that are essentially protein factories. These are responsible for synthesizing all of the proteins in a cell, which we already know have very important roles for the cell, so it makes sense that they are found in all living cells regardless of type. So we've just been talking about the ways in which cells are similar to each other, the structures that they have in common. And now we're going to move on to talking about how cells can be different from each other. And we'll start this discussion by talking about the two major categories of cells that are recognized by biologists, which are prokaryotic cells and eukaryotic cells. Prokaryotic cells are the category of cells that are smaller and simpler of the two. They do not contain any internal structures that are bound by a membrane, also called organelles. The word organelle literally means a tiny organ, and prokaryotic cells do not have these. They're very simple. Prokaryotic cells are all single-celled microscopic organisms. And what this means is that every organism that is a prokaryotic organism is only made of a single cell. They're not made of multiple cells like animals or plants, for example. Some examples of prokaryotic cellular organisms include the bacteria E. coli, Escherichia coli, which is what you see in the upper picture here, and the bacterium Streptococcus pyogenes, which is, as the name suggests, the cause of strep throat, which is what you see in the lower picture here. So all types of prokaryotic cells are tiny microscopic bacteria. While they are simple and there's not a whole lot to them, there are a few structures that prokaryotic cells have that we need to talk about. One of them is something called the nucleoid region. The nucleoid region is the area in the prokaryotic cell where the genetic material is located. There is not a special compartment inside of the prokaryotic cell that holds its DNA, and this is something that sets prokaryotic cells apart from eukaryotic cells. Instead, prokaryotic cells simply have a region or a space inside of their cytoplasm where they store their DNA, and that space is called the nucleoid region. Many, but not all prokaryotic cells can have a structure called a flagellum, which is like a tail hanging off the back of the cell made of protein, and it's responsible for propelling the cell through its environment by rotating like a propeller. Prokaryotic cells also have something called a cell wall, and a cell wall is a layer located outside of the cell membrane that is used for extra protection and maintenance of the cell's shape. This material, or this layer, is made up of a material called peptidoglycan. And in addition, outside of the cell wall, some bacterial cells, some prokaryotes, can have another layer of material, this one made up of carbohydrates, called a capsule. Not all bacteria have a capsule, whereas all bacteria have a cell wall, but the capsule can offer additional layers of protection and can actually make a bacterium more virulent or dangerous to humans because it is more able to ward off our immune system defenses. So here we come to another applied checkpoint. The antibiotic penicillin interferes with the chemical compound peptidoglycan. Which structure in a prokaryotic bacterial cell does penicillin destroy? So that completes our discussion about prokaryotic cells. As you saw, they're very simple, and now we're going to move on to eukaryotic cells, which we will see are much more complex. They are also larger in size than prokaryotic cells, and they contain many internal structures that perform specific functions. These cells can be either single-celled microscopic organisms or large multicellular organisms. Both exist in the world of eukaryotic cells. Some examples of the wide variety of organisms that are composed of eukaryotic cells include mushrooms, elephants, amoebas, humans. Everything you see on the screen here is made of eukaryotic cells. And in fact, eukaryotic cells are so diverse that we divide them into different subcategories. So here are the 4 different types of eukaryotic cells: plant cells, animal cells, fungi, and protists. And protists are like single-celled microscopic organisms, such as amoebas, paramecia, stuff that you might see growing in pond water. But these guys are not bacteria. They're actually made of eukaryotic cells. So now let's talk about the structures that can be found in eukaryotic cells. And as we do this, we are going to be mindful of the fact that not every eukaryotic cell is alike, and that plant cells may be different from animal cells, which may be different from fungal cells, etc. One thing that eukaryotic cells can have, although not all of them have it, is a cell wall. Prokaryotes we saw had a cell wall, which was responsible for protecting the cell and maintaining its shape. And in eukaryotes that have cell walls, this is also the function of their wall. The cells of plants and fungi have a cell wall. And in fact, when Robert Hooke was looking under the microscope at that sample of cork tree, what he was seeing in that honeycomb shape was actually the remnants of the cell walls of the cork tree. Animals and most protists do not have a cell wall at all. One thing that all eukaryotic organisms have, however, is a nucleus, and this is a significant feature that sets them apart from prokaryotic cells. Because as we saw, prokaryotic cells did not have a special compartment for their genetic material. The nucleus is a special compartment bound by a membrane called the nuclear envelope, which is dedicated to storing and protecting the DNA. Within the nucleus, there is a structure called the nucleolus, which is the specific location in the nucleus where the ribosomes are created. So as we can see in this figure right here, the larger, lighter pink structure is the cell at large. Within that cell, the darker pink structure is the nucleus. And within that darker pink structure, there is a purple circle, and that is the nucleolus. Eukaryotic cells also have something called the endoplasmic reticulum. The endoplasmic reticulum is made of membrane, just like the outside of the cell is. It is a single continuous layer of membranes with many folds, and it can have 2 separate forms. In this image right here, we see the rough endoplasmic reticulum. Now, the image is pointing to these layers of membrane here, and those are the rough endoplasmic reticulum. You may also be able to see in this image that those layers look like they are a little bit textured and bumpy. And the reason why they look like this, the reason why they have this rough texture, is because they are studded with ribosomes. You may remember that ribosomes are a structure that's common to all cells because they have the very important function of creating the proteins for the cell. So the rough endoplasmic reticulum is responsible for processing the proteins that are made by the ribosomes. That's what it does. The other form of the endoplasmic reticulum is called the smooth endoplasmic reticulum. And in this image right here, you may be able to see that while it does show layers of membrane here, those layers of membrane are not textured and rough like the ones in the other picture. And that's because smooth endoplasmic reticulum is not studded with ribosomes. It does not have them. Therefore, its function has nothing to do with proteins. Instead, it contains enzymes that are responsible for synthesizing lipids and steroid hormones. Another structure that eukaryotic cells have is something called the Golgi Apparatus, also known as the Golgi body. The Golgi Apparatus is kind of similar in appearance to the endoplasmic reticulum in that it is a series of membranes or sacs that you can see pointed out here, but the function is different. The Golgi apparatus is where the cell wall material is synthesized. It's also where proteins and lipids from the endoplasmic reticulum are sent in order to be chemically modified so that they can perform their functions. The Golgi apparatus spits out the molecules that it modifies in these pockets of membrane material called vesicles. Vesicles, which you can see here, are defined as pockets of membrane that bud off of organelles or the larger cell membrane to transport materials. What you can see going on in this microscopy image right here is a vesicle forming from left to right, where the membrane forms a pocket, that pocket deepens, and then eventually it pinches off to form a continuous and self-enclosed vesicle. And that vesicle can be sort of like a little escape pod that moves materials around the cell or dumps them outside of the cell. For example, lysosomes are a specific type of vesicle that contains digestive enzymes for breaking down large particles, and the lysosomes play a very important role of cleaning out the cell of waste materials. It takes large waste particulates, breaks them down, and then merges with the cell membrane in order to dump them outside of the cell. Another structure found in eukaryotic cells is the cytoskeleton. Cyto, the prefix meaning cell, and then followed by skeleton, of course, means that this means the cellular skeleton. Cellular skeleton, or the cytoskeleton, is a network of protein fibers that run throughout the cell. And just like your skeleton does for your body, the cytoskeleton supports the shape of the cell. It keeps the organelles, or the internal structures such as the Golgi and the endoplasmic reticulum, in fixed locations within the cell so that they're not just sloshing around everywhere. However, the cytoskeleton can also help move materials around the cell by pushing them in a particular direction when need be. In this microscopy image right here, the cytoskeleton material is the stuff that is stained in neon green, whereas the nuclei of these cells are stained in blue and the walls are stained in red. Next up, we have vacuoles. Vacuoles are essentially water sacs. They store water and materials that are dissolved in that water, and they are only found in certain types of cells. They're only found in plants, fungi, and protists, not animal cells. So our human cells do not possess vacuoles, but other types of cells do. The central vacuole is the specific type of vacuole found in plant cells, which is called the central vacuole because it is so large. It is responsible for storing large amounts of water, which allows plant cells to regulate their shape, size, and water content. And in this microscopy image right here of a plant cell, the central vacuole is this giant space inside of here that looks like it's full of nothing, but actually it's full of water. The central vacuole can sometimes be so large that it takes up to 90% of the internal volume of a plant cell. The other type of vacuole is called the contractile vacuole. And this type of vacuole is found in protists, which are the single-celled microscopic organisms that are not bacteria, but rather a eukaryotic microbe. The contractile vacuole is responsible for pumping water out of cells that take on too much. These types of organisms, these protists, always live in watery environments, for example, in ponds. And so it is common for them to take on too much water and then need to pump it out using their contractile vacuole. And it's called a contractile vacuole because that pumping action causes it to contract. As we can see going on in this animation right here, the contractile vacuole squeezes down and pumps water out of the protist cell in order to regulate the water content. Next we have chloroplasts. So chloroplasts are another one of those organelles that's only found in certain types of eukaryotic cells. They're found in all plants and some protists, but they are not in fungal or animal cells. Chloroplasts have a very important role in the metabolism or energy processing of the organisms that possess them because they perform a process called photosynthesis. Photosynthesis is a chemical reaction that generates sugar molecules using the energy of light. This is how plants obtain energy. Another interesting thing about chloroplasts is that they contain their own set of DNA, which is separate from the DNA that is found in the nucleus of the cell. We're going to circle back to that fact later and talk about why that is the case. But before we do, we're going to talk about one more organelle, and that is the mitochondria. Mitochondria are also an important organelle for energy processing and metabolism because this is where another cellular process called cellular respiration takes place. Cellular respiration is the series of chemical reactions that break down sugar molecules in order to obtain energy for the cell. Mitochondria are found in virtually all eukaryotic cells, and like chloroplasts, they too contain their own set of separate DNA from the DNA that's found in the nucleus. So why is that the case? Why are mitochondria and chloroplasts special in the sense that they have their own set of DNA? Well, this gets to something called Endosymbiosis Theory. The theory of endosymbiosis says that chloroplasts and mitochondria specifically originated when one large cell engulfed another smaller cell, and the two went on to develop a symbiotic relationship where the smaller cell provided energy for the larger cell and the larger cell provided protection for the smaller cell. And many years down the line, it is now the case that the chloroplast and the mitochondria, which used to be free-living, probably bacterial cells, are completely codependent on the eukaryotic cells that contain them. There's a lot of evidence for this theory, including the fact that these organelles contain their own DNA, which suggests that they used to be separate living organisms. They also contain their own ribosomes, which we know is a characteristic of all living organisms. They are also approximately the same size as a prokaryotic cell. And they are also susceptible to antibiotics, which suggests that they were probably once free-living bacterial cells many evolutionary histories ago. This bacterium that you see right here is a type of bacteria called Gloeomargarita lithophora, which, if you look at the DNA that is found in this type of bacteria and you compare it to the DNA that is found inside of chloroplasts that are in plants, this bacteria's DNA is very similar to the to the DNA that is found in the chloroplast. And so this bacterium is believed to be a very distant cousin and the most closely related existing species to the chloroplast. So that completes our discussion about eukaryotic cells, and we're going to tackle a few checkpoint questions here to round out this lecture. In the first one, I want you to compare and contrast prokaryotic and eukaryotic cells by describing two things that they have in common, as well as two things that they do not have in common. In the next checkpoint question, I want you to tell me three structures that plant cells possess which animal cells do not possess. And in addition to that, tell me what are the functions of those three structures. Once you finish these last two checkpoint questions, then you are finished with this chapter.